Recombinant DNA technology is something special that, as opposed to classical breeding that we used to do to improve crops or animals. So in classical breeding, normally you take one type of, you breed of a dog or a horse, and then you breed with another type, and then you create an offspring that has the traits of both the parents. But in such breeding, there's always a limitation of a species. You cross between two species, but if you cross two different things like a tiger and a lion, you may create a liger, but that will have characteristics of both tiger and lion, but they become sterile. They are not fertile to reproduce more offspring. You'd always go back to the parents and to reproduce. The beauty of recombinant DNA technology is that it can cross this species barrier. You can cross even kingdom barrier or domain barrier, and you can take a specific gene that codes for a specific trait, then take it from the bacteria and express it in humans, or take a gene from human to express insulin and put that into a bacterium and make the bacteria secrete insulin. So this type of technology was not available until we understood what DNA is, how it makes RNA and protein, and how we can regulate that gene expression and also make many recombinant DNA that were non-existent before. So in this recombinant DNA technology, you need to understand what are the toolkit materials available to you, as well as what are the techniques that you need to know to make this possible. So we are going to first look at the tools that are enzymes, basically, that manipulate DNA, cut, combine, and so on, and also DNA-based tools that are called vectors that would take the DNA from one organism, transfer it to another organism. So the enzymes, you need to have enzyme that can recognize a specific strand of DNA and cut it in the middle, called as restriction endonucleases. Also, you need to have enzymes that can connect the DNA, called ligase, and those type of enzymes are essential to create recombinant DNA. But in addition, you also have enzymes that make DNA, called DNA polymerase, and enzyme that make RNA, RNA polymerase, or enzyme that would make DNA from RNA, called reverse transcriptase. All these enzymes are very critical in this recombinant DNA. The next set of tool is called as the vectors, or cloning vectors. So this is not same as the map vector. And the cloning vectors are vectors that are double-stranded DNA, and they are derived from bacterial plasmids, called the plasmid vectors, or derived from bacteriophages, called bacterial viruses, a viral-based vector called phage vectors. So these are essential to take DNA from one organism, insert it into the vector, and replicate in a host cell. So these are essential to create recombinant DNA, copy recombinant DNA, transfer recombinant DNA. The vectors essentially contain an origin of replication that is critical to replicate the DNA. They also contain a selection marker called ampicillin selection marker. And thirdly, they have a multi-cloning site where you can insert foreign DNA. So once you understood the tools, the enzymes, and the vectors, then we need to move on to techniques. So the techniques are very, very essential. And these techniques are rapidly being developed as we speak. So it is very important for you to understand at this stage what these techniques do, or what these techniques are used for, than how they work. It's good to understand them, but it takes much longer time of exposure or experience to fully understand how these techniques work. The simple techniques such as gel electrophoresis can be used to separate the fragments of DNA once you cut with the restriction enzyme. Or PCR, polymerase chain reaction, is used to make multiple copies of the gene. Or DNA sequencing is used to understand or elucidate the base sequence of DNA. So all these techniques try to understand what they are used for. Okay? Once you know the techniques, then the next thing is to understand how are they used. Application side for either humans or plants or animals. So understand how these are applied either to develop new diagnostic kit or to treat a particular disease. Can you give a recombinant vaccine or a recombinant treatment? So these are the different ways that you can apply recombinant DNA technology. So there are so many things that are available right now. You can create a recombinant vaccine, or you can create a therapeutic treatment to treat blood clot for stroke, or you can also diagnose diseases before a fetus is born. And you can also think of cases where you can understand, where we came from in terms of studying the evolution of human or studying the evolution of other organisms. So this recombinant DNA technology has so many different applications. You may need more than a semester or to study all these applications. Try to have a bigger picture. Lastly, for this chapter, we need to understand what are the safety regulations involved, what are the ethical implications involved if you are going to manipulate the gene in the form of DNA and then introduce to another organism, and that is critical. So the ethics is more based on the society. With the safety, it comes to the scientific safety of using recombinant DNA or a recombinant virus. Who makes these rules and who enforces these rules is good to have a general understanding. And then if you look at the overall, this chapter talks about the tools involved in recombinant DNA and the techniques that are employed to understand a particular process or to understand a particular nature of the molecule. And then you come to applications where you utilize these tools and techniques to develop a diagnostics or a treatment or general understanding of the different organisms, and they all come under applications. Finally, look at the safety involved in this new technology as well as what are the ethical implications.